Paul says I can start talking anytime, so I'm going to start now. I know a number of you. I'm Carolyn Lawrence Dill. I've worked in this community for many years. I'm now the Associate Dean for Research and Discovery in the College of Ag and Life Sciences at Iowa State. And it is my pleasure to introduce our speakers this afternoon. I have sort of an uncommon introduction for our first speaker, and I'm going to speak from the perspective of a co-PI on a grant. I'm a co-PI on AG2PI, that's the Agricultural Genome to Phenome Initiative. The URL is here at the bottom, www.ag2pi.org. What AG2PI is trying to do is to develop connections across communities, and specifically across the crop and animal communities, livestock communities, in the genome to phenome space. We're working to develop a vision for genome to phenome research, to identify research gaps and opportunities, to foster community solutions to challenges and gaps that we identify, and then to disseminate those findings. And so we do that through training workshops, field days, conferences, and seed grants. And the way that I know Fiona is sort of, I've known her for forever, it feels like. But for the introduction here, the way that we came to know that Fiona was particularly interesting was because of the seed grants. So the people that got seed grants from AG2PI, we've been querying them and surveying them, and they're probably sick of being queried and surveyed on what they're doing. But one of the things we did was to ask them who they were working with that were among this community of people. And so once you figure out who people are working with, you can start to figure out what a network would look like, how these people are working together. So the one network that you're looking at right now, the question was, prior to joining the team, meaning your seed grant team, who did you learn from? And you can see a big group at the top of plant biologists. You can see a network at the bottom of animal biologists. And then there are a bunch of individuals that have not really been working with people that have seed grants for AG2PI. The nodes are people, and the connections between those people are the edges between the nodes. So then we asked, since joining the team, who do you learn from? And so clearly it changed. The interesting thing is how it changed, and that is that the plant and animal communities really came together based on this orange dot that's kind of in the middle of this one person. That one person is Fiona. So Fiona is an interesting animal scientist for us to invite to this meeting to learn what it is that she's doing and what her efforts are in animal science. I printed out just a small thing to let you know where she's working. She's a professor in the School of Animal and Comparative Biomedical Sciences at the University of Arizona, Tucson. She's a founding member of the Ag-based databases, and I can't not tell you this because it's interesting and I want to know what this is. She teaches a course called Living Dangerously. Fiona.
Okay. I'm not sure what to do after that introduction. Okay, that's okay. You can hear me okay? Okay, let me know if that sound is okay. Okay. All right, thank you everyone for inviting me. As despite what Carolyn tells you, I'm very rarely allowed to speak to plant scientists and even less to plant breeders. So I'm really excited to be here. I've got a really ambitious topic, so let's see what happens next. Okay, so I don't need to tell anyone in this room about the incredible achievements in agriculture. We have a long history of genetic improvement, and what I want to do is focus on the right-hand side of this picture, milestones in plant breeding. I want to focus on the genomics, the genetics, and everything that comes from that, and really sort of try to talk about opportunities for plant and animal scientists to work together and hopefully inspire some more collaborations for Carolyn's network there, right? Okay, so of course, all of this is moving towards precision breeding. Precision agriculture is an even bigger topic. It involves all that Internet of Things, all that wonderful technology that we can apply to farm management practices. I'm going to focus in on the, like I said, the genetics and the precision genetics. So I'm talking about things like all the genetic selection technology and genotyping, the gene editing, the phenotyping, and underpinning all of this, the foundation that makes it possible for us to do any of this, of course, the data analytics. So as I'm going through this, I'm going to try and point out opportunities for cross-kingdom collaborations. So what I'm looking at here, and I'm having even trouble seeing this slide, there we go. So looking at, I want to talk about support, what we can do as a group to support genome annotation efforts, talk about what we can do to develop the phenotyping technologies, develop the data analytics capacity that we need, and of course, with that, it's absolutely essential that we bring our education and training systems along. So the first example I want to talk about is the FANG project, the functional annotation of animal genomes. And this really, I guess, started with the 2008 release of our animal genome blueprint, our USDA blueprint. This USDA blueprint really did focus in on 3 areas. It wanted to see that precision genomics, that science to practice, that very applied genomics and genetics. We wanted to see the discovery science, all that foundational information that we need to build these applications, and underneath that, the infrastructure. So we're talking about the resources, the tools, and most importantly of all, the thing that I think often gets forgotten, the human expertise, the human capacity to do all of this, okay? So, like all of the other ENCODE projects, the goal of this is to catalogue all of the genetic elements, find all of the functional elements within the genome, not just the genes, but all the regulatory regions. So unlike the other ENCODE projects, the human ENCODE project, the mod ENCODEs that you've seen done, we knew there was a couple of things we were going to do differently. First of all, of course, we're interested in different species. We wanted to start looking at different species, and we wanted to do this in an organised way so that we could facilitate the coordinated, facilitate the comparative studies. So what we have here is a set of species, and for each species, we're going to pull out a set of tissues as comparable as they could be. Of course, these tissues were going to be tissues that were important to our production traits. So unsurprising things like muscle, adipose, immune function tissues, okay? That sounds easy, but it was of course much harder than that. And then we were going to do the genetic assays. So we're thinking the gene expression data sets, the non-coding RNAs, find all of those, start looking at methylation and ChIP-seq and all of those types of assays, and just magically put it all together. How easy could this be? At about the same time we were talking about this, this paper was published. This is a call for the PENCODE, the Plant ENCODE project. And I bring this up mostly because I want to talk to this group with the perspective of what we've done and what we've learned and what we would advise you not to do if you're going to develop an ENCODE project. But most important of all, to tell you why maybe you should revisit this idea and think about it. Now, of course, it's absolutely possible for individuals to go out, grab their species, their tissues, their developmental stages, do their assays, and then somehow hope that all this data will one day get integrated. But it's extremely hard to do. And part of this, of course, is the problem we have because these are all new assays. It involves new reagents that have got to work in our species of interest, and that is not something that agricultural scientists take for granted, like medical people do, okay? And then we've got to, so having this idea of doing this in a coordinated way, I think really helps the entire community and brings the entire community together. So our first thing that we had to do was to come up with an agreed upon set of hopefully manageable species, tissues and arrays. And this is the first step that you're going to upset people with, okay? Because clearly I want my things of interest in there, but so does everybody else. So that's something you've got to think long and hard about. How do we set these goals out, okay? Of course, what we want to do is have a manageable set so that we can focus not just on getting that data out for people to use, but developing the expertise, developing the capacity to do all of this, okay? We need a common agreement for data release and making sure that that data gets out into a place where someone can find it and reuse it or even just look at it. With all of this comes the capacity to do the data analysis for all these different assays that we're now employing, right? So thinking about what works in our species, what breaks when we try to use it in our species, what can we use? And this idea of working through this software and coming up with workflows that at least we have one workflow for analysis that we know would be okay. And then, of course, bringing all this data together. So this is an example from the FANG website showing the data samples being submitted, and the most important reason why I put this up was because of this. This is one of these things that comes out of a collaborative and coordinated project like this. This is an information on how to get all your data sets out to the public archives, which archives, with documentation about exactly how to go about getting it into the archive. Now, if anyone's ever tried to do data submission, it's not a trivial process, and just having this as a resource for our community has been just incredible, okay? So what have we learned? And keep in mind, this is a project that's ongoing. I'm sure we've got plenty left to learn. Collaboration is key. I can't stress that enough. It's not one of those collaborations where you get specific aim 1, you get specific aim 2, you get specific aim 3, see you back here in 3 years. You need to have continual conversations, you need to be continually working together and telling people what you're doing, okay? You need to engage the public resources really, really soon in the process. You know, I'm talking about NCBI, Ensemble. They have the data management skills and they're used to dealing with big, messy, complicated data sets, okay? We didn't have Ensemble on board when we started. It took a few years. They later became our data center. And since that's happened, it's been amazing to see the progress that's been made. Of course you have to involve the funding agencies, that would be nice, but also involve the people who are the data consumers. Who's going to be using this data? What are they going to be doing with it? What do they expect to see? Okay? Transparency is essential, and as researchers we're really good at telling you about the positive results, terrible at telling you about the negative results. For a project like this, it's really important that not only do we know what's been done, we know what has not yet been done. We need to know what has worked for these assays, but also what has not worked and what has not even been tested yet, okay? Mission creep is a killer. Focus, focus, focus. We need to really focus in on that core set. Of course, everyone has their species lines, breeds, tissues, developmental stages, and assays they want done. But if you can focus on that core set, what you're doing is you're providing that expertise and reagents and all of that resource that other people can then use to study these conditions and data sets that they're most interested in, okay? And then, of course, and I put this down here because it's you know, individual data is not interesting to anyone, not even a bioinformatics person. It's the information and the knowledge that you get when you bring this all together and look at it in some meaningful way. I'd like to tell you that we know how to do that. I'll keep you posted. So there's opportunities here for cross-disciplinary projects thinking about genome annotation and supporting genome annotation. And if you ask a bioinformaticist, this is the answer you get. We need to sort out our software, our data analysis. We need to have workflows that actually focus on the work part of workflows, okay? We need software that we know that works, and we put this together and we can come up with expectations that lead to community standards and best practices. And this all sounds so terribly boring. And if you're going to put in a research grant on this, this is not going to get you your novel and innovative and exciting research grant, okay? So this is the kind of thing I think that we've got the USDA research programs, the multi-states and national programs, they can do this well. We've got USDA and NSF that do these research coordinated networks that could really help with something like this. And of course, NSF is developing capacity in cyber infrastructure grants. So there is research out there for this. I put education and training there. I always come back to education and training. It's no good building the best workflow in the world if nobody knows how to use it, okay? We have a group of people who are doing this, who are experts at data management. This is the Ag Bio Data group. This is people from all the agricultural databases, and they think about this kind of, well, they think about data management, they think about integrating data and what it looks like and all that boring stuff that you don't want to have to think about. They think about it. Wouldn't it be nice if we had some group like this doing this for the actual analysis? That would be awesome. I keep pointing it up there. Okay. Carolyn stole my thunder on this one, but I'd like to talk to you about the AG2PI, the Agricultural Genome to Phenome Initiative, okay? So, at least from my perspective, our new animal genomes blueprint came out, it was around about 2018, and at the same time, it really focused on the genotype to phenotype, and round about the same time, there was a big push to get phenotyping into the Farm Bill. We managed to do that, and from this, the AG2PI was born. Okay, tell me your events later. Okay, I'm sure it was harder than that. The really fun, cool thing about this is it combines the plant and animal communities. This is awesome, right? This is what we can use to start developing some of these really cool ideas, okay? The idea here is that we want to develop these phenotyping technologies, and of course, as we develop these phenotyping technologies, we need to think about how we're going to link this back to our genotype technologies, okay? Of course, we want those applications for agriculture, and I think this phenotype and tying this in, getting away from the pure genomics out into what's happening in the real world is this way to get to this application and deliver on the promise of genomics. But it's going to need to be high throughput phenotyping, okay? And I think that putting this together and building this really builds the next generation of all of our genetic tools. So our genetic selection, our gene editing, all moves to the next level if we do this right. So I think one of the problems that we have here is that phenotypes are way more diverse and way more specific. And what I mean by that, of course, is the phenotypes are very tailored to specific industries. Every industry, every crop grower has a particular phenotypes that they are interested in. The good news is that we have a history of defining and collecting traits and phenotype data. So we know we can do this. We need to scale it up, okay? Now we're thinking about linking our genotype data, our genetics data. Now we have to start linking it to all of this phenotype data that we're hopefully going to get. And that means thinking about how do we manage, process, analyze image data, audio data, video data, and how do we put it together with our genetics data? So what you're talking about here is going from bioinformatics to data science. And this can be kind of freaky because I think we're barely getting a handle on some of our bioinformatics. And I'm standing here telling you, no, look in the rear view mirror, we're moving ahead, we need data science, okay? But I do think there's incredible opportunities to be found in this work. Obviously, we need more work in defining our phenotypes and our methods for collecting them. But, you know, one of the things that we've done with genomics up until now is we've really been siloed based upon our species, our genomes, our industries of interest. But for phenotype data, I think we have to build those bridges across the silos, because I think what we've got to see here is when we're thinking about how we're going to manage our phenotype data, it might be best thinking about it in getting together in groups based upon people who are using phenotype data maybe collected from the same systems, for example, drone data, or people who are trying to analyse the same types of data, people who want to do image analysis. Yes, you might be doing image analysis in crops and pigs, but image analysis is the fundamental problem you're trying to solve. So I think this is something where we have these cross-disciplinary opportunities to really build out on this and work together as a group to do this. Of course, I've said it before, I'm going to keep on saying this, we need that expertise to be able to bring together now not all of these diverse genetic data types, but now even more data types, so all of those phenotype data types. And we've got to have data science expertise to manage that. Now, we have our national projects for animal genomes and crop genomes. These are people who have come together to solve these problems and work on these things. Many of these people are also involved in our ag biodata. So we have precedents for figuring out how to do this, how to come together as groups to solve these problems. Okay? I'm going to talk about education and training because I don't think we can not talk about it. We know our employers, the employers of our students, want this data-savvy workforce. And many of us are involved in education. We're teaching the next generation of students. Add on top of that the training that we've done for bioinformatics, where we've involved industry groups as they want to update their training, and they talk to us about what needs they want for our graduating students. And what we really need to have is this data analytics as the foundation of the education for our animal and plant science students, okay? And I get into trouble when I say this, but what the heck? I know we can't, you know, I talk to people and I say we need to have bioinformatics now, data analytics, data science in our programs. And I get told that we have an already crowded curriculum, we can't possibly put another course in there. And they're right. And I agree, we shouldn't be putting another course in there. What we should be doing is putting pieces of data analytics into every single course in our program that we offer. Because a one and done single course on bioinformatics is not going to get you the traction you need, okay? That's something where you check the box and you move on and you get your degree and hopefully never speak of it again, right? But if you we have students who are passionate about animal science, passionate about plant sciences. They came for this reason, to learn about this. They need to have this data analytics in their program. I've got on the side there data carpentry, software carpentry. These are the educational programs for learning more about data sciences and how to analyse your data sets. I will tell you, everyone I talk to around the US as we talk about these problems, very few of our students will do data carpentry, software carpentries. Why? Because what they care about is the plants and the animals. They didn't sign up to do computing. What we need is the context for them. By putting pieces of data analytics into this coursework, we're telling them this is reproductive biology, this is plant stress biology, whatever it is, and yes, you do need data science to analyse this. And you put it in every course, and as my collaborator says, this is how you teach them to eat their vegetables. You just keep slipping it into the meal at every opportunity, okay? So I'm going to sum up by really taking a step back. And one point I really want to make is I think we have a lot of opportunities to work together. And even deeper than that, I think we have the philosophy, the state of mind where we are really after a lot of the same goals, okay? We've had, we're much closer working with each other than we are working with, in my example, medical science. I've worked with medical science, and it blows my mind every time. You know, every software tool you pick us up works. It's like, it's so uninteresting compared to what we do on a daily basis. Everything works. You need more data? Sure, I'll go get you more data. Wow, you're making this easy. I think we've got a lot more in common in the way we think about things and the things we do to get our research done, okay? We have decades of developing well-characterized lines and breeds that we're using. We have decades of experience collecting QTLs and genetic markers. We're used to thinking about multiple species, looking at multiple species. Pangenomes don't scare me because I've been talking about lines and breeds for as long as I've been doing this research, okay? And most importantly of all, and I'll finish on this point, just when I talk about philosophy, I think the thing about agriculture is anyone who's been doing this work in agriculture, we've had 10,000 or more years of just doing more with less, okay? It's what we do. And I'm going to stop on that note. That's my final slide, and I'm happy to take questions. And Carolyn has promised that no one's going to throw things at me. So remember that. Thank you.
Recording in progress.
Are we doing the next one? I can't actually see anything here. They've just gone to get more tomatoes, haven't they? I know. Oh.
Hi. So I was wondering, do you have any thoughts on how you're going to keep like analytical framework longevity when it comes to this ag to bio project? Because I think one issue that comes up a lot is there will be an initiative that has like really great analytical tools for a while, but then as soon as like funding runs out, it you know all kind of goes to dust.
Thank you for setting this question up for me. This is wonderful. Sustainability, right? And this is something that anyone working with the databases, we get this, you know, because it's a lot to set up a resource, whether it's workflows, whether it's databases, and then how do we balance continuing as a community to pay for that every year versus doing additional research. And I think we need to look to the NSF because I think they've done this well, and you are very fortunate because plant science has conduits into NSF that animal science as a general rule doesn't. I will say I've thought a lot about this, and one of the things I've been thinking is that we now have the technology in a lot of ways to transcend databases and to take a lot of the pain of the maintenance and keeping things updated out of this. We have things like GitHub, which people can work on developing software and continue to work on it as long as the community's interested. So I think the cost of the infrastructure for these resources can be really, really efficient in ways that it couldn't 5, even 10 years ago. So I don't know if that specifically answers your question. It's something I think about a lot.
Thanks again. I really appreciate your presentation. And again, thank you for the plug for AgBio data. I mean, this is something near and dear to my heart because the data that everybody here is producing is integral. It needs to be given to the next generation. So when you publish your data, the question that I'm going to bring to you is, how did you get it? How that documentation about submitting data? Like, you said it was something really important. Like, how much work did it take to do this? Like, what is needed in that? I can give a webinar every month about how to submit data to the database I work for, and no one will do it.
Yes.
So
I share your pain.
help me.
I think there's a couple of things here to unpack. First of all, you can build these resources. Will they come? Of course, some will. You're right. You're just picking a top layer off, perhaps. I think where you really start getting to this, and the interesting thing that I've seen since FANG started is, and it coincided with the USDA getting serious about data management plans, where it became more, it moved from being a suggestion for a proposal to, you must have a data management plan. And within 1 year of being on review panels, I see reviewers who aren't me, by the way, they put in a data management plan. It only had 3 lines. They didn't think this through. This takes time and effort. That has been an amazing transformation to see. And people are even doing the point now where they're going and saying, these people, they collect the money, but I don't see any data going into these repositories. And reviewers talk about that. So I think the two places where we have the power to make this change are for, as reviewers, for papers and for proposals. Expect to see it. And this comes back to this idea of give them the tools to make it easier so that they can do what you're asking them to do. I share your pain, I really do. Okay?
Thank you very much.